We have 3 more to come that I think will all. Please pay attention to the emails when they go around and show up. And some of you have gone to all of them, and that's great. But I encourage everyone to do that. The second announcement I want to make is I forgot to do an introduction of the new member last week. Chris Kozak, you just walked in. I saw you. Stand up, please. So Chris is a new postdoc in my lab working on the CCGP together with Rory and Fred and me and others. And he's going to be working on the bioinformatic analysis of the mammalian genomes that we are sequencing. And Chris sits in the office that Fred is in at the end, back behind Rory. Are there any other announcements before I turn it over to David to introduce today's speaker? Becca.
Oh, yeah, there's another new postdoc here, and who's working with the CCGP.
Please stand up so everyone can see you. Sorry, it's our tradition to embarrass everyone.
Hey, and technically in Elon's lab, right? That's she's sitting over in Bogert.
Well, welcome to the MVZ, and we hope you spend a lot of time here. Jim.
I'm going to introduce Lake Olson from
thank you.
Mammologist at the University of Alaska in Fairbanks, very well known for her studies on everything from shrews to marmots and the world.
And one of each of those that Jim has collected. So.
And you're here taking advantage of our collection and looking at some specimens. So.
Your weather.
And our weather. Thank you. Anything else? Okay, I will turn it over to David to introduce today's speaker.
Hello, everyone. It is my pleasure to introduce the next in our series of MVZ student invited speakers, Professor Martha Muñoz, assistant professor in Ecology and Evolutionary Biology at Yale University, and also, if I'm correct, the assistant curator in herpetology at Yale's Peabody Museum as well. Martha did her PhD at Harvard University with Jonathan Lassos, and fun fact, Jonathan Lassos did his PhD at the MVZ. So I see this as an opportunity for us to get a chance to meet and interact with our extended MVZ academic family. So welcome. Martha's laboratory group research mainly focuses on the major topic of understanding the tempo of evolution, specifically how different phenotypes evolve at different rates, taking advantage of primarily 2 study systems, Caribbean Anoles and also Appalachian salamanders. Yes, and so lastly, as I think it's some of you may know, well, when the MVZ students nominated, and then when I reached out to Martha to invite her for this talk, it turned out our own Becca Tarvin already has invited her for IB seminar, which will be happening in this building, not in this room, but downstairs tomorrow. So I just wanted to say thank you to Martha for taking time for another day of seminars and meetings. We're all very happy to have you here. You can all please help me welcome Martha.
Thank you, David. That was absolutely lovely. What a lovely sentiment to be considered part of the MVZ family. I definitely share those sentiments, and Jonathan and Chris Schneider, who's another mentor of mine, are both MVZ graduates, so it's very special to be here. Thank you so much for the invitation, and I'm also really jazzed to try this out for the first and maybe only time ever, maybe tomorrow as well. Yeah, so let's get into the research. Today I want to talk about some of the emerging work in my lab, and to do that I really want to start by emphasizing really the scope of my work, and my work is fundamentally at the broadest level motivated by the fact that evolution proceeds unevenly. Sometimes adaptation and speciation can be exceptionally rapid, as if it were driving along evolution's autobahn. But in other cases, evolution can actually be quite slow, as though it were stuck in New York City or San Francisco, as I discovered yesterday, at rush hour. That was a very long ride. Okay, so let's talk about diversity. We can think of 3 major axes of diversity. We can think of species richness, ecological diversity, and phenotypic diversity. And you can sort of consider these to be independent knobs that can be tweaked and turned. At one extreme, we have adaptive radiations. These systems characterized by high species richness and also high ecological and phenotypic diversity. A couple of charismatic and classic examples would be Caribbean anoles. Those are going to be featured today, but also Malagasy bangas and many, many other systems. And you can think of adaptive radiation as a special case of evolution in which adaptive divergence keeps pace, so to speak, with speciation. In my opinion, just as interesting is sort of the flip side of this equation and non-adaptive radiations. Lineage is often also characterized by really high species diversity, but also characterized by a paucity of phenotypic and ecological differentiation. Some classic examples would be several sublineages of plethodontid or longless salamanders, like the slender salamanders and the woodland salamanders that I'll be talking about later today. And of course, there are actually many ways, thank you to Chris Martin and Emily Richards for this incredible graphic, because it's super useful, many ways to carve up species richness, ecological and phenotypic diversity, with plenty of biological systems filling the gamut. Now, I'm intrigued by these evolutionary disparities, right? So for example, to what extent are our inferences about the patterns of evolution dictated by the specific types of traits that we examine? And likewise, from where do these disparities arise in the first place? Well, one of the major ways in which I broach these questions is by studying one of evolution's key architects, which is behavior. Through their behavior, organisms are endowed with the capacity to modify the selection that they experience, in turn having major consequences for patterns and rates of adaptation and speciation. One of the more classic takes on this is perhaps the idea that behavior should drive evolution. One of the major figures behind this idea was Ernst Mayr, who spent a lot of his career traveling around the tropical and subtropical regions of the world and inferring the mechanisms creating all of the incredible biodiversity of birds. And he postulated that that diversity was in no small part due to behavior. Behavior allows organisms to exploit new resources within their environments or enter new environments altogether, in turn exposing organisms to selection and driving evolution. The flip side of this perspective is that behavior should actually slow evolution down. Organisms that preferentially select microhabitats that are within their ancestral range have the power to buffer themselves from natural selection, in turn limiting phenotypic divergence. These ideas were originally formulated in the mid-20th century, in particular by Charles Bogert and a few others. So in honor of his pioneering efforts, you may know this as the Bogert effect, but it's also known as behavioral inertia. And I usually study these interactions between organisms and their environments and how that impacts evolution in a couple of vertebrate systems, in particular reptiles and amphibians, and in particular with respect to hydro and thermoregulation. And today I'll be focusing on two major systems that my lab focuses on: Caribbean Anolis lizards and plethodontid or lungless salamanders. Okay, so today I'd really like to start by just giving you an overview of what I've learned about the Bogert effect over the past 10 years and how we can deploy the Bogert effect to understand these disparities in evolution. Then I'm going to shift gears and start giving some empirical examples from ongoing work. I'm going to start with Anolis lizards and describe how patterns of morphological and physiological evolution can actually tell us different stories about that radiation and how the Bogert effect specifically plays into that. Then I'm going to discuss some aspects of climatic niche evolution in plethodontid salamanders and discuss how that might relate to their physiological diversity as well. Can everyone hear me okay? Okay, awesome. Okay, so like I said, the Bogert effect describes this restraining role of homeostatic behaviors on evolution. Organisms can preferentially select environments to which they are already well adapted. This has the potential to shield organisms from environmental extremes, limit exposure to selection, and slow evolution. Now, I first became interested in the Bogert effect as a first year. Who are the grad students here? Any first years? Awesome. Okay, so, no pressure. I first became acquainted with the Bogert effect as a first-year graduate student. In particular, I read a phenomenal paper by Ray Huey, Paul Hertz, and Barry Sinervo. And when considering the Bogert effect, I think they took it just one little conceptual leap forward and made the following statement. Under the Bogert effect, the response to selection will be less than expected slowing rates of evolution under natural conditions. It's just one sentence in a paper, but man, did it strike a chord with me. And it really stuck with me because it sets up an interesting hypothesis, which is that the traits and lineages that can be behaviorally buffered from selection should evolve more slowly than the traits and lineages that cannot so readily be buffered from selection. So I became sort of fascinated with this idea and wanted to explore it more. At the same time, I was also reading a lot of George Bartholomew, a very renowned comparative physiologist of the 20th century. And in one of his papers, he made also the following statement. He also thought about a lot about the ameliorating role of behavior. And he said, this is a direct quote, "Behavioral avoidance, not physiological adaptation, is an organism's primary response to an environmental challenge. This point is elementary, but it is by no means trivial." Emphasis on the last part. By no means trivial is true, Bart, really true. And after about a decade of picking apart the Bogert effect from several different dimensions, it really does turn out that the devil is in the details. So let's talk about those details. And in order to describe what I've learned about the Bogert effect, I do have to take you back in time a little bit and give you a little bit of the origin story of my interest in this phenomenon. And that story begins on the Caribbean island of Hispaniola. This is the island that is comprised of Haiti to the west and the Dominican Republic to the east. It is itself a very topographically complex and large Caribbean island, divided into two paleo islands, each with its own mountain range. The Cordillera Central in the northern paleo island, and the Sierra de Bauruco in the southern paleo island. All of this topographic complexity translates into a lot of thermal variation across the island. Because it's over 3,000 meters, it does get pretty cold at high elevation. Now, near sea level, it's as warm and balmy as you might expect of the Caribbean, but at high elevation, it gets really cold. In fact, it's not uncommon to see frost, even in the summer, on the highest peaks of Hispaniola, which for a friolera like me is a very big departure from what I thought I'd be doing when I said I was going to the Caribbean for research, which for which I was completely underprepared and, like the lizards, thermoregulating to the best of my capacity. But I had actually excellent company on the summits of these mountains in the form of Anolis lizards. Anolis armori on the summits of the western mountains and Anolis shrivi on the summits of the eastern mountains. And these two montane anoles are themselves part of a broader radiation of so-called trunk ground anoles, anoles that are perched from about here to the ground from the Caribbean island of Hispaniola. Now, I spent a lot of time exploring the regulatory behavior and thermal physiology of these lineages. For example, we spent a lot of time just out in the field measuring body temperatures, looking at environmental temperatures, and estimating patterns of thermal regulation. We also measured a couple of key physiological traits, for example, the critical thermal minimum and the critical thermal maximum. In ectotherms like these anoles, these define the acceptable thermal boundaries for activity. Temperatures above and below these, they lose the ability to locomote. So they're really important ecological traits. So we measured these in all of those anoles. So you may also know this as cold tolerance and heat tolerance. I'm going to use CT min and cold tolerance interchangeably, likewise for CT max and heat tolerance. We also brought some animals back to the lab and exposed them to a thermal gradient to sort of get some information about their preferred temperatures. These are all just standard physiological traits to sort of define niche width and niche position from a physiological perspective. And to summarize, very sadly, many years of my life in 3 sentences, what we discovered is actually very little differences in these species, if any at all. If you look at their body temperatures, it only drops by like 1.5 degrees across 2.5 kilometers of elevation. If these same lizards had been behaviorally passive with respect to the environment, it should drop by more than 10 degrees. In fact, if you give me a body temperature from a cybotoid anole, cybotoid being this clade of trunk ground anoles, I couldn't tell you the population or species from which it was gathered. The same is true for heat tolerance. The same is true for the preferred temperature. The only trait that seems to vary is cold tolerance, which becomes progressively stronger with elevation, with those high elevation anoles being up to 7 degrees Celsius more cold tolerant than their low elevation counterparts. So this is a lineage, just putting this all together, of incredibly intense contradictions. But that conundrum made much more sense once we got more information about the temperatures available to those lizards. So what I've shown here are daytime environmental temperatures across 4 sites that vary dramatically in elevation across Hispaniola. And what you can see, perhaps unsurprisingly, is that there's a lot of thermal variation during the day, right? And importantly, a lot of thermal overlap among sites. Maybe it doesn't get quite so warm at high elevation or quite so cool at low elevation and vice versa, but overall, there's a lot of thermal overlap. And when we superimpose these lizards' preferred thermal range on there, we see that all sites on Hispaniola offer the opportunity for thermal regulation within their preferred range during the day. This thermal heterogeneity is the raw material for thermal regulation. This is what organisms are using to play with in their environments. At night, by contrast, the thermal environment compresses, only about 5 to 10 degrees Celsius variation at any given site, and importantly, it gets progressively cooler with elevation. Correspondingly, the clinal relationship between cold tolerance and elevation that we observed makes intuitive sense. But we can take this a bit further and revisit now the Bogert effect. Given the asymmetry in the efficiency of buffering behaviors, we might predict that the Bogert effect is actually stronger on the upper features of the thermal performance curve, like heat tolerance or preferred temperatures, than it is on lower features of the thermal performance curve, like cold tolerance. In essence, is there a Bogert effect occurring here? And in fact, there is. Not only does cold tolerance evolve faster, it evolves an order of magnitude faster than features of the upper end of the performance curve. And there have been lots of studies to sort of do similar things in other systems, and this seems to be a generally robust pattern. Lower physiological limits tend to evolve faster than upper physiological limits. But now let's explore the Bogert effect from a slightly different dimension, because there's a lot of reasons why thermoregulatory capacity could vary. And one of those is just, thermoregulation itself hinges on the spatial distribution of temperatures. Even 2 daytime environments can differ a lot in how costly they are for thermoregulation, simply depending on how much canopy there is. In general, edge/open habitats provide a lot of sunshade structure. Organisms can play with that to thermoregulate. By contrast, closed canopy forests tend to be more thermally homogeneous and generally cooler. So this sets up the possibility that if there's a Bogert effect at play, that rates of evolution should be fastest for organisms occupying environments that are costlier for thermal regulation, following, and it should be slower in organisms that occupy environments that are less costly for thermal regulation. And in fact, that's exactly what we observe across multiple systems, not just anoles. In many systems that we've examined, this seems to be robustly the case. Organisms that occupy environments costly for thermal regulation, their physiology tends to evolve faster than lineages occupying environments that are actually easier or less costly for thermal regulation. Okay, now, just to make the Bogert effect that much spicier, let's add another ingredient to the mix, and let's revisit another quote from George Bartholomew. He's going to pop up a lot in this talk. From the functional point of view, of course, an animal is indivisible, and physiology is not in any sense an isolatable component of an organism. Very true. If physiology is defined as the study of vital functions, I like that definition too, it becomes inseparable from morphology and behavior. Now, we've already sort of been hinting or flirting with the idea that behavior and physiology are related in an evolutionary sense, but Bart's point about morphology is incredibly prescient. First, behavior, morphology, and physiology in a general sense can't be separated. Organisms can choose any microhabitat or site that they want, but their size, shape, and color will determine the thermal consequences of that choice. Secondly, and also importantly, thermal regulation itself might require a shift in substrate use or perch use or some change in how organisms utilize resources. And in so doing, whatever that behavior is may have additional, potentially contrasting selective influences on other aspects of the phenotype. Or to borrow another quote from Ray Huey, Paul Hertz, and Barry Sinervo, homeostasis in one trait can be achieved only by non-homeostasis in other traits. So returning to our example of the cybotoid anoles, that's exactly what I observed. At low elevation, the lizards perch primarily on tree trunks, but at high elevation, they've become almost completely boulder dwelling or saxicolous. The reason is rocks are the only perch warm enough for thermal regulation to be effective. Trees are simply put too cold. So in other words, the Bogert effect and behavioral drive might be to a certain extent interdependent phenomena. And I'd certainly unpack this more in tomorrow's talk. But the point here is that behavior, morphology, and physiology are kind of locked together in a delicate evolutionary dance, no piece separable from the rest. Okay, armed with this information about the Bogert effect, now let's revisit the adaptive radiation of anoles. Now, adaptive radiation, we know, is a phenomenon that theoretically includes divergence in physiology, behavior, and morphology. But much, if not most, of what we know about adaptive radiation really centers around morphology. This is a bit of a limitation because to really understand how adaptive radiation proceeds, we need a better grasp of how multi-trait evolution proceeds. And this is a bigger question than just asking, for example, if morphological and physiological evolution differ in radiation, although that's certainly important as well, but it's bigger than that. It's, you know, to what extent do different traits evolve independently? How do trait axes saturate sequentially or collectively? What information do we miss if we take an exclusively physiological or an exclusively morphological perspective? Or put differently, is there something bigger and more insightful about how evolution works by combining these axes that is greater than the sum of the parts? Okay, to answer this or to think about this, I'm going to focus on Caribbean Anolis lizards. This is a highly diverse radiation that is widely distributed throughout the tropical and subtropical regions of the Western Hemisphere. So was there a question? They are exceptionally diverse in the Caribbean, where more than 150 species can be found. Now, this is the radiation that for sure has been historically predominantly studied from the perspective of morphology. In fact, the adaptive radiation itself, is, at least in the Caribbean, is somewhat defined by the convergent evolution of different ecological specialists called ecomorphs. I've shown a few here from each of the major islands just to show you how convergent these assemblages are, right? So our understanding of Anolis is really also a restatement of ecomorphology. But also surprisingly, switches between ecomorph categories explain a relatively small proportion of overall Anolis diversity. To explain more of that diversity, we have to also think about evolution along different axes, and one of the most important has actually been with respect to temperature, both with respect to diversification across thermal macrohabitats, for example, across elevation, but also with respect to thermal microhabitats, for example, across the sunshade axis. So how do we piece together these different dimensions of the radiations? Well, theory posits that during adaptive radiation, trait saturation should occur sequentially, that is in a specific order. And the idea here is that by sequentially saturating, we can probably result in greater species packing by minimizing ecological overlap among close relatives. For example, in Cuba, there are places in Cuba where you can get 11 species of anoles co-occurring. That's more anoles than even occur on the entire island of Puerto Rico. So that's a lot. These ideas have certainly been worked out in Anolis lizards. Specifically, these ideas were formalized in the 1970s by Ernest Williams, shown here. And he postulated that anoles first diverged in morphology in body size and shape to permit ecomorphological differentiation, and that secondarily, ecomorphs diverge in ecophysiology predominantly along the sunshade axis. He did most of his work in Puerto Rico, and most of what we know about this hypothesis is from data on Puerto Rico. And there on that island, this tends to play out. Species in the same ecomorph tend to diverge secondarily in sun and shade use. Those that utilize open habitats tend to have higher heat tolerance and a higher optimal temperature for sprinting versus those that occupy more close canopy habitats. So I guess the entrance to this project, we were sort of wondering, is physiology in a general sense just as deterministic in this radiation as it has been for morphology? You know, do the same patterns that we see on Puerto Rico bear out across the Caribbean? And if not, why might that be? And there's a couple of ingredients that may actually make this story more complicated than it's been told. The first is not all Caribbean anoles fall into ecomorphs. we have a couple of morphologically unique one-offs, which we call the unique anoles, which simply means they have no correlates on any of the other islands. These unique species are only found on the two largest islands of Hispaniola and Cuba. It includes some true weirdo bizarros, like Anolis barberi, a leaf litter anole that for me is much more phenotypically reminiscent of a Burkessia leaf litter chameleon than anything we might think of as an anole. It also hops. Oops. And plenty of other anole bizarros with no correlates on any of the other islands. We know exceedingly little about these organisms, about their morphology and ecology, and even less about their physiology. Secondly, the story of anole ecomorph evolution has largely centered on low to mid elevation assemblages. High elevation environments on the Caribbean, however, have received comparatively less attention, even though diversification across elevation provides a clear substrate for physiological specialization. Okay, so here comes this study, and that's exactly what we sought out to do. We were going to focus on Dominican anoles. We were going to figure out and compare patterns of morphological and physiological evolution, and then compare that to what we know from Puerto Rico. And the work I'm about to describe is being led by Brooke Bodensteiner, a PhD candidate in the lab holding a super rare species of anole, Anolis landestoyi. That's why she's cheesing so hard. She caught it too, super rare. And she's done a phenomenal job with this. Okay, so like I mentioned, we focused on Hispaniola for a couple of reasons. One, every Caribbean ecomorph can be found on Hispaniola. There's also several morphologically unique species. Species do vary in canopy use, and they've diversified across elevation. They've got all the ingredients we're looking for. Now, the morphological data that we worked with was body size, or SVL, snout to vent length, and hind limb length, two features really strongly associated with Anolis ecomorphology. The physiological traits that we worked with were heat and cold tolerance. And here's where it's a little emotional, because there I am during my PhD getting CT max from a trunk ground anole from Hispaniola. And here's Brooke in 2019 doing the same thing on a crown giant for CT min. So we combined our data sets, and in total, we had morphological and physiological data from all Dominican anoles, 29 species in total, representing every major lineage on the island, all the ecomorphs, and several unique species. So the sampling was pretty good. Okay, now there's many ways in which we could try to address this question about ecomorphological versus ecophysiological specialization, but let's start simple. Let's ask, to what extent is ecomorphology a signal of ecophysiology and vice versa, right? Are ecomorphs also physiological specialists? What I'm showing here are just our morphological traits, hind limb length and body size. And when, not surprisingly, the ecomorphs cluster in morphological trait space. This is just a rehash of ecomorphology. This is nothing new. What I want to do now is ask a slightly different question and ask whether morphology corresponds to any repeatable signals in canopy use. So these are the same morphological traits, and now we're just wrapping species based on canopy use, and not really, right? No clear signal. Let's do a slightly different exercise, and now let's look at how physiological variation is partitioned among ecomorphs. Now we have cold tolerance and heat tolerance on the X and Y axes. And we don't observe any major pattern. What you can see is that there's no clear signature of being in a certain ecomorph being associated with having a certain physiology. When thinking about Puerto Rican anoles, so far this is entirely consistent with Williams's scenario and what we observe on Puerto Rico. Species in a given ecomorph don't necessarily also converge in physiology. Now, when we look at how physiological variation is partitioned among canopy use classes, we do seem to find that open habitat species have higher heat tolerance than species in closed canopy forest. Let's probe that a little bit more deeply. On this plot, I'm showing you elevation on the x-axis and cold tolerance on the y-axis. What we found is that both for open habitat and closed canopy species, cold tolerance decreases with elevation. And importantly, canopy use class imparts no distinct signature on either the slope or the intercept of this relationship. By contrast, heat tolerance also decreases with elevation, but it really only does so in those closed canopy species. In species occupying open/edge habitat, we see no pattern at all. Going back to our scenario about the Bogert effect, this makes intuitive sense. Those closed canopy environments are much more thermally homogeneous, limiting opportunities for thermal regulation. This is sort of what we expect. But also recall that those differences in thermoregulatory capacity might be associated with evolutionary rate differences if the Bogert effect is at play. And in fact, that's exactly what we observe. Those anoles that are found in closed canopy habitats have a faster rate of heat tolerance evolution than species occupying those edge open habitats. Remember that thermal heterogeneity, as particularly that afforded by edge and open habitats, facilitates thermal regulation, and under the Bogert effect should slow evolution. That's exactly what we find. Okay, but I want to probe this from a different dimension still. So bear with me. So what I want to do now is ask a slightly different question, because we're sort of hovering around this idea that the Bogert effect is stronger for heat tolerance than for cold tolerance. And we're also finding that it might even be stronger in open habitat species than in closed canopy species. So there's a lot to take apart here. One way, so in general, we expect a species physiology to reflect its thermal environment. That trait-environment relationship can be disrupted through regulatory behaviors. And we're predicting that that disruption is stronger for CT max than for CT min. We can put this idea to the test by using the Slouch Framework. This framework assesses the pattern of adaptation of a continuous trait like CT min to a continuous predictor variable. In this case, we use mean annual temperature as a general proxy of the thermal environment. There are a couple of parameters we're interested in. The first is phylogenetic half-life. This tells you how quickly a lineage gets halfway to its phenotypic optimum. A lower value indicates rapid adaptation. The trait rapidly catches up to any changes in the environment. Longer phylogenetic half-lives, approaching the total length of the tree, by contrast, indicate like a protracted lag in adaptation, that the trait isn't tracking the environment to such a degree. We can also think about stochastic variance, how much noise there is about that phenotypic optimum. Under the Bogert effect, we would expect phylogenetic half-life to be longer, and potentially that species are adapting to a wider adaptive zone if the Bogert effect is at play. So we started with CT min, and what we observed, again, expecting a very strong relationship here, is actually that phylogenetic half-life is essentially 0 or close to that, meaning that as soon as organisms enter a new environment, cold tolerance adaptation rapidly follows. This is effectively another way of restating the clinal relationships that we were observing earlier. By contrast, CT max also seems to track the thermal environment, but it does so with a much longer phylogenetic lag and about a wider adaptive optimum, reflecting this sort of Bogert effect of sort of disrupting or decoupling the trait from the environment. We also discovered when we did the same test, so now we're looking at how morphology adapts to mean annual temperature. So how does morphology respond to the same predictor that we use for physiology? And we discovered like a weak signature of body size evolution in anoles, such that high elevation anoles are a little bit smaller than their counterparts near sea level. But this wasn't particularly strong, and we discovered no relationship whatsoever between residual hind limb length and the thermal environment. So kind of consistent with, so far with Ernest Williams's hypothesis, so far these patterns are kind of bearing out. Let's take a slightly different tack here. We know that because this is an adaptive radiation, morphological evolution should be quite rapid, right? So we already know this from Caribbean anoles. Now let's just add physiological traits to the mix. Is rapid evolution an equal morphological phenomenon in anoles, or do the physiological traits keep up? Well, morphological evolution definitely outpaces heat tolerance evolution, but the real star of this show, in my opinion, is cold tolerance, because it evolves up to 44 times faster than any of the other traits. Or put differently, to the extent that we would say that anoles are an adaptive radiation characterized by rapid morphological evolution, we have to also include cold tolerance in that definition. And just because Jonathan's not here and I get to tease him, I'm going to suggest, maybe provocatively, that anoles are actually a physiological adaptive radiation with a hobby for ecomorphology. Okay, let's add one more test and now really hit the crux of Williams's hypothesis and discuss the temporal sequence of trait evolution. Now, recall that under Williams's hypothesis, morphological traits are expected to saturate early in the lineage's history and physiological traits later. This is the temporal sequence that he proposed. We can use the node height test to sort of put these questions to the test. Now, we expect trait contrasts if there's no temporal pattern to evolution, we don't expect there to be any relationship between trait contrast and branching time. By contrast, I shouldn't say contrast here, when the contrasts are steeper at deeper nodes in the trees, we detect an early burst of evolution. And when the contrasts are steeper towards the present, we detect, by contrast, a late burst evolution. I've also used contrast 5 more times since trying not to use that word. But effectively, you can detect early versus late burst evolution in different lineages just based on the sign of the relationship between the contrast and the branching pattern. Okay, so under Williams's hypothesis, we expect an inverse relationship for morphology, a positive relationship for physiology, reflecting early and late bursts respectively. For morphology, no signature whatsoever. In fact, traits seem to saturate progressively with time. What about physiology? Well, we do detect a late burst of physiological evolution, but it's actually in cold tolerance and not in heat tolerance. What does that mean? Well, in terms of limiting ecological overlap among close relatives, it really doesn't, because any given species at a site will be equally cold tolerant, right? So it's not permitting fine scale thermal habitat partitioning in the sense that Williams meant. That doesn't mean it's not important for the radiation. Clearly it is. Anoles diversify across elevation, and in doing so, cold tolerance evolution rapidly follows. But perhaps the more interesting outcome of this is that the patterns observed on Puerto Rico with respect to ecomorphological and ecophysiological evolution really don't seem to bear out when we look at Hispaniola. And those patterns seem to kind of fall apart. And that's interesting because anoles are otherwise such a paragon of deterministic evolution. So what gives? Well, perhaps in a general sense, replaying the evolutionary tape with respect to ecomorphology results in more deterministic evolution because the adaptive substrate to which morphological specialization responds is fundamentally repeatable across islands. The same structural supports that are found on Jamaica are found on Cuba, Hispaniola, and Puerto Rico, right? By contrast, these Caribbean islands definitely differ in a lot of the features that dictate physiological evolution: topographic complexity, overall size, temperature, humidity. All of these factors differ between islands. And this just creates fundamentally different ways that anoles carve up space. So for example, on Hispaniola, we have, because of the incredibly high elevation of this island, we get a lot more parapatric divergence across elevation, things you just don't observe in Puerto Rico. These are all very closely related species. They're in the same ecomorph, but instead of partitioning the sun-shade axis, they're actually differentially distributed across elevation and separated by an ecotone. And Cybotes is less cold tolerant than the 2 montane endemics. Likewise, the historical geology of Hispaniola matters here. For example, Anolis chlorocyanus and Anolis celestinus are ecomorphologically and ecophysiologically indistinguishable. Instead, they are separated by the biogeographic boundary called Mertens line that separates the northern and southern paleo islands. So the presence of multiple paleo islands is also something to consider in this context. So where to next? Well, our goal is to really take this project pan-Caribbean, and I'm sort of fascinated to see how this plays out because, like Hispaniola, Cuba is divided into multiple paleo islands. Like Puerto Rico, Jamaica, Cuba are kind of altitudinally shallow. So there's a lot of ways in which these major features that clearly seem to dictate physiological evolution differ across the Caribbean. I'm sort of curious to know, how do these patterns bear out? You know, how do we integrate these? And are there general rules by which physiological diversity arises in this radiation? And are those rules just different from how ecomorphological variation arises? To do this, I'm collaborating with Luke Frischkopf and Luke Mahler, and we have this project to go across the Caribbean and do exactly that. We did get to go into the field last summer. I do want to share this because it's been a while since we could go out into the field. So here I am up a tree in Puerto Rico deploying a copper sensor to measure temperature. And we're going to do that across different habitat types in the Caribbean, across different canopies. We sort of want to know to what extent is a structural niche also a thermal niche, and use that information to sort of calibrate the patterns that we find. Okay, I don't have a ton of time. Until when do I go?
You got about 10 minutes.
Oh, I do have 10 minutes. Oh, I can probably finish in 7. All right, Challenge accepted. Thus far, I've been focusing on adaptive radiations, the special case of divergence in which adaptation keeps pace with speciation. Just as interesting to me are these sort of anomalies, these non-adaptive radiations, organisms in which there's a lot of species richness, but a paucity of ecological and phenotypic divergence. And I want to focus in particular on one of the most classic examples of non-adaptive radiation, which would be woodland salamanders in the genus Plethodon. These are North American salamanders. You do have some on the West Coast. And I guess, okay, they're really cool. So it always hurts a little bit to say this, but if you see one Plethodon, I mean, it's not to say that they're all the same, but if you've seen one Plethodon, you've kind of seen them all. They look very similar to each other. I still love them though. But you know, they're kind of rad for many other reasons. First of all, you know, they achieve such Plethodontid salamanders in general achieve biodiversity hotspots outside of the tropics, which for herps is super rare and unusual. They also have really interesting species richness and elevation relationships, and they're just a fascinating clade to work with. But here I want to focus on a slightly different question, which is, yes, ecomorphological differentiation is rather limited in Plethodon, but what about ecophysiology? Is this a dial that these organisms have tuned to sort of, that can explain some of their high species richness? And in order to get at that, this is actually a really big question, we sort of had to start small and on the ground, literally and figuratively. This project started in the field, and the work I'm about to describe is led by Vinny Farello, a former postdoc in my lab, in collaboration with Joseph Weta at Virginia Tech and Don Miles at Ohio University. And this project really set out to ask, first and foremost, what are the hydric and thermal patterns of microhabitat use in Plethodontid salamanders in North America? Vinny's study location focused on the Appalachian Mountains. Look at species richness. That's wild, right? That is wild for North America. If we zoom in on his sampling site, you can see that he visited a wide range of places at different elevations. And this was actually a tour de force. Everywhere he went to, he measured the temperature and relative humidity at site of capture for salamanders. He did this over the course of 3 years for 26 species of Plethodontid salamanders, resulting in 3,000 individual observations. So it really was a Herculean effort. What did we do with those data? Well, at the same time that he was out in the habitat, you know, measuring where the salamanders were, he was also doing surveys of random habitat to sort of understand what was available to the salamanders. We need that information to know whether and by how much they might be hydro or thermal regulating. This allowed us to partition our data in many different ways that allowed us to sort of take a fully organism independent perspective of the thermal and hydric environment and subject it to greater behavioral filtering until we result at like direct measurements of temperature and humidity at the site of capture. Okay, so we analyzed these data in several different ways. I'm going to share two with you today. And the first was by examining how phylogenetic signal differs when subjected to greater behavioral filtering. Now, phylogenetic signal in and of itself is just a statement of the statistical dependence of phenotypic traits on relatedness. When signal is low, relatedness has really low explanatory power for the distribution of phenotypic variation. I've created a dummy phylogeny and some dummy data, but this is an example of low phylogenetic signal. Close relatives are no more close, phenotypically close than let me rephrase that. Close relatives do not share similar phenotypes, put more simply. Now, keep in mind, we expect close relatives to look the same or similar, but by how much is something that definitely differs among lineages and traits. By contrast, when phylogenetic signal is high, you can see a lot more clustering such that close relatives have actually quite similar trait values than distant relatives. Now, to be clear, phylogenetic signal reflects a lot of different things. So we're not really as interested in the amount of signal per se, but rather how the magnitude of that signal changes when subjected to greater behavioral filtering. So that's exactly what we did. What you're going to see here are a series of phylogenies that show the distribution of the hydric and thermal data as we apply more behavioral filtering. And what we observe is that when you look at the direct site of capture, that's when we get the strongest amount, the highest amount of phylogenetic signal in the data. Sorry, this is for hydric use. Same pattern when we look at thermal microhabitat use. There's absolutely no phylogenetic signal to the data when we look just at the temperatures available to these organisms. But then when we zoom in on the temperatures that we actually observe them at, we get a lot of phylogenetic signal. There's a lot of phylogenetic structure to the data. And for those of you familiar with George Bartholomew, talk about being caught in a phylogenetic trap past evolutionary adjustments. But anyway, so just to sort of, so now when we combine at the direct site of capture, the hydric microclimate and the thermal microclimate, we get a pattern that makes intuitive sense, which is that organisms are picking combinations of temperature and moisture that reduce evaporative water loss relative to their environments. And they do this through two different tacks. Some species prefer it warmer, but then they also prefer it wetter. Some species prefer it cooler, but then they also prefer it drier. And these combination of features reduces evaporative water loss more than expected based on a random sampling of their environments. When we zoom in on woodland salamanders, this genus I was talking about, we find the same pattern. In fact, microclimatic preferences seem to be structured along major phylogenetic breaks in this lineage, with species in the glutinosis group preferring it warmer and wetter, and species in the Cinereus and Whirlii groups preferring it cooler and drier. Now, we also explored this using the Slouch approach from before, because again, we should anticipate that those microclimate preferences track the prevailing macroclimate, but through the Bogert effect, we might actually anticipate phylogenetic lags in this tracking. So we explored this, and what we discovered was actually that phylogenetic half-life was much lower for hydric microclimate use than for thermal microclimate use. This suggests to me that this is the primary, this is the feature of primacy for these organisms. They're adapting their hydric physiology to their environments. Any differences that we might observe in thermal preference, by contrast, may simply reflect the fact that thermal physiology is tracking hydric physiology to minimize water loss. So of course, where you'd want to go from here is matching this up with experiments. And so that's the natural next step of this work. And that's what we're doing. So like all of us, this work was severely held up by the pandemic, but this talk actually catches us at a really interesting moment. Megan Gate, a postdoc in the lab, recently went with Julia and Henry, two graduate students, out to the Appalachian Mountains, and we've got 150 salamanders in the lab from over 20 species that we're subjecting to a variety of physiological experiments. And I should mention that this work is being conducted in collaboration with the amazing Eric Riddell from Iowa State. And that's where we are. We're sort of, we're like in the middle of it. It's happening right now. And we're measuring a whole slew of features, thermal traits like heat tolerance, cold tolerance, preferred temperatures, water loss rates across a variety of temperature and moisture treatments, metabolic rates, and we're also looking at morphology. To give an example of how literally like this is happening right now, this is a photo from last week. I actually tweeted this last week. This was my station, this is Saul's, and we were just measuring heat tolerance in salamanders. So hopefully within a few years, I'll have much more to share with you about this. But spoiler alert, they're not very heat tolerant. That's what we discovered. Okay, I don't know if that was 7 minutes, but I think I almost made it. Before wrapping up, I really just want to thank everyone in the lab, because all of the work I presented today really reflects their combined effort, and they are amazing. And with that, I want to thank you and take any questions that you might have.
Time for questions. Thank you, Mark.
Yeah, Becca.
Yeah, about the salamanders, curious what you know about where they lay their eggs and if that has anything to do with it.
That's a very good question. I don't know if it's necessarily just about reproduction. No, because the sampling was sort of across the entire year. But we never, you know what? That's a very good. So what if we partitioned hydric and thermal microhabitat use between reproductive and non-reproductive periods? Will we detect a stronger? It's a good idea. Thanks, Becca. Yes.
Yeah, so I was wondering, with the Anoles in particular, what's known about how much they can acclimate to different cold regimes, and how do you disentangle that?
Oh yeah. So yes, it can. So this is something we worried about with our Anoles. So what we did was basically do like a long-term acclimation treatment to them. And we were only really able to modify CT min by about a degree. So I think if you accounted for any kinds of, well, and we also did our sampling in summer when physiological differences are actually minimized across elevation. They should be even steeper in winter. So this was a conservative estimate of physiological differences, but nonetheless, plasticity matters. We redid our analysis sort of correcting for that, and the rate difference does go down a bit, but not that substantially. Yeah. Chris.
Fantastic talk, as always.
Thanks, Chris.
Really amazing. But I guess I was wondering with your Anolis section, so if you expand to other aspects of physiology, like the Woodland Salamanders, I think you'll find similar patterns, like olive grade or something.
You know, the beauty of the thermal work with anoles is that you can do it all in the field. And doing the hydric work right, you really have to bring them back to the lab. You can sort of hack it in the field, but it's never as satisfying. I think it does matter because the ecotone separating the forest from, okay, so open habitat, fantastic for thermal regulation. Probably, I mean, a lot more convection, a lot more evaporative water loss. Those species should be, you know, there should be some hydric signature of physiology that compensates for this thermal difference, right? Or to put Bart and Huey back on the spot, like non-homeostasis, homeostasis in one trait probably requires non-homeostasis in another. I'd love to get at that. It's on the docket, and I have hypotheses, but you're right. Oh, sorry. Oh, I guess. Hi, Paul. May I take this call? Okay, and then Jim, and then okay.
Oh, I'm sorry. I didn't want to interrupt anyone else. I can't see the rest of the people. Well, thanks for revisiting the Anoles. It was fun to see them again. I would caution you about the easy to measure parameters and what may be more important. CTM and CT max have been around for a long time and can be discredited a lot of the time. Depends on how you measure them and so on. And there's some question if they're relevant. You can measure preferred temperature in a gradient pretty easily. But I think what we have found over the years is none of these tell but maybe the more important story of what these, I'm thinking of anoles now, of what the anoles really do. They don't need their preferred temperature. Some of them need their preferred temperature to function. Others don't. And the CTM, the max, I think is very clear. You cook or you don't cook. CT min is much more complicated. And the question is, how do they function? And on Puerto Rico, you can see this where lizards with the same preferred temperature, some can be active when it's much cooler and others can't. So it's easy to measure certain things, but they may be a little too simplified.
Well, thank you for that comment, and I share a lot of the same thoughts.
Can you repeat the gist of what he said?
So he mentioned that CT min and CT max and T pref to an extent tell you something about the organisms, but the ecological relevance can be much bigger. So I think that was the nature of the comment.
Close enough.
Much appreciated. Thank you.
You're welcome.
Jim.
Yeah, I'm just curious if there's much variation in the elevational ranges of the Anolis species that you're studying and whether or not that factors into the rates that these species can respond to different temperature and stuff. What plays a role in there?
They do differ. So two species, Anolis longitivialis and Anolis whitemani, they can only be found till about 450 meters. Now, their TB is no different than the other species, right? So it's not, you know, again, I couldn't tell you a species based on its TB, T pref, or CT max. So everything I can't address the second part of your question, but the first part, yes. There's a lot of altitudinal differences. Then Anolis marcannoi and Anolis straumi are from sea level to about 1,000 meters. So you have a lot of species. In fact, most species in this radiation start around sea level, and the question is how high do they get? Some get a little higher than others. Anolis cybodes is essentially found island-wide, except at the highest elevations where it becomes parapatrically replaced by Anolis armorii in the western mountains and Shrivi in the eastern mountains. Those are the only two cybotoids that I'm aware of that don't have any low elevation counterparts at all.
Yeah, I guess the nature of my question was, are these high elevation lineages in particular, are they specialized for that environment, or do you have some that go from low to high, and what would happen if you were comparing low and high elevation individuals?
Gotcha. Yeah, they're definitely specialized. They're the only anoles found at those elevations. And they have a couple, yes, their heat tolerance and T-prep and TB are no different, but they do differ in other things besides CT-min. Their behavioral signal structure has diverged. Okay, step back. They occupy these really open environments where there's big warm boulders. This also exposes them more to predation, right? So they've changed their signal structure and they compress more information into shorter displays. So that's also different. They're also warier. The escape distance is like 3 times longer at high elevation than low elevation, I think reflecting, you know, the dangers or the stronger predation pressure, especially from avian predation, which does exist at high elevation, even though there's no snakes up there. They also, the females retain their eggs in the oviduct longer than at low elevation, and the reproductive season is somewhat truncated relative to low elevation. So you have and anoles only lay 1 egg at a time, which is just not the best strategy, but it's what they do. And at low elevation, they're like boom, 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 you know, yolking follicle egg, and they just go back and forth throughout the entire reproductive season. And at high elevation, they retain them for weeks longer, and then they lay them. So there's a major fecundity difference as well between high and low elevation. Those lizards perching on boulders are also darker colored. It could be thermoregulatory or it could be substrate matching, you know, because they perch on boulders. So they're specialized in a whole slew of ways that make them really different from the other cybatoids. It's just not evident in their heat tolerance, T-prep, or TB. Michael.
As you were presenting this, I was thinking how the patterns might be different in endotherms versus ectotherms, and also in nocturnal versus diurnal animals. Can you comment on that a little bit?
I consider endothermy the ultimate Bogert effect. But yes, so nocturnal and diurnal. Interesting you asked. So the original studies by Charles Bogert and Raymond Cowles in the early 1940s, which established thermal regulation as a thing, we didn't even know, or it wasn't even formalized until the 1940s, which is wild. They did this study in desert in the Coachella Valley here in California. And to their surprise, desert lizards and snakes had TBs that were much lower than anticipated given the heat of the desert. This sort of primed them to think that desert reptiles were not like exceptionally heat adapted, although this had been a common viewpoint up until that point. The only major difference between those reptiles was deal activity, with nocturnal species exhibiting lower core temperatures and voluntary thermal maxima than diurnal species. And this has been followed up over the years. Ray Huey, but others, John Weens, plenty of people have sort of broached this question, and it seems to also still play out as well. I would expect, all else being equal, that the Bogert effect is stronger for nocturnal organisms than diurnal ones, but there's noise there. Like Caroline Williams has some really elegant work looking at thermoregulation in crickets, right? And they're nocturnal, and they do a lot more than anoles might. Anoles are inactive at night, right? So I don't know, it'd be interesting. Broach that dimension. Great question. Oh, I skipped you. I'm so sorry, please.
Thank you so much for the people. I'm really sorry. It seems that a lot of the quantification of your patterns downstream is going to be dependent on the robustness of your phylogenetic features and like on species definitions and all that. So I was wondering, how do you how do you manage that relationship between your morphological data and the robustness of your?
Are we being recorded? I'm not gonna say who this quote came from, and it wasn't me. It was a major figure in the field. The anole tree sucks, is the provocative statement. Yeah, that is a limitation, and other vertebrate systems, I think, have sort of built more robust phylogenies than we have currently available for anoles. Not to say there isn't a tree and it isn't good, but it sort of could be better. And that's something that Jonathan and colleagues are working on right now. To my understanding, they're doing whole genomes and they're using that to sort of build a much, much better anole tree. I look forward to those days. Jim.
I'm just kind of following Michael's question and thinking about Eric Riddell, who you mentioned, because he was here as a postdoc.
That's right.
On the Cornell project, and the difference in the response of birds and mammals in the desert to increasing climate change, both decrease in precipitation and increase in temperature over the last century, has been birds that get hammered. Mammals have not been, because their behavior, not just because their nocturnal and diurnal differences, but the behavior of mammals is to sequester during the daytime in the heat, in refuges, but also physiologically to adjust to torpor so they can serve resources. And so both of those are, as I understand it, are Bogert effects, but they have different outcomes for the organism, and they're both endothermic.
Fascinating. Yeah. Now we should talk about Eric with that and revisit the details of his project. It's very cool.
Yeah, he's a great.
Oh yeah, no, I mean, I sought him out for this. He's brilliant. I love his work. I was going to build on something you said, which is, rising temperatures, drier environments sounds like a bad recipe for any West Coast salamanders as well. Yeah.
One more question.
Oh yes, please.
Hi, yeah, great talk. I was really pleased to see the last bit of it on non-adaptive radiation. So this is going to be a bit of a cynical question to be answered over the course of all your careers. But I gave a talk here 17 years ago, and someone asked me if there was such a thing as a non-adaptive radiation. My answer wasn't nearly as good as yours, but your answer seems to be leaning towards no.
Well,
And
okay,
I just
let me walk that back a bit.
But this is why it's going to have to be answered by these folks here, is that we use these words like hidden and cryptic to basically whitewash our ignorance, because we have to figure out what might be under selection and what might represent adaptation. I'm just wondering if you think there are any classic non-adaptive radiations that will stand the test of time.
Oh dear. First, I just want to revisit the previous point. I don't think that every non-adaptive radiation is an adaptive radiation waiting to be discovered necessarily, just by definition. I think, I don't know, allopatric lineages present promising systems in which it might be the case that there's truly non-adaptive divergence. But I think a lot of this dates back to a lot of hardlining perspectives on whether divergence is purely allopatric or purely ecological. And I think the reality is somewhere in the middle, right? And whether we say this falls enough, this is, if we're separating black and white or red and blue, this is enough to say it's an adaptive radiation versus this one isn't. I think that's more qualitative, but I do think it's going to be generally true. And that's why I say hidden in quotations, because it's hidden in plain sight, right? It's this sort of channel of diversification that organisms can utilize that gets comparatively less attention. It might be the case that as we subject more lineages to these sorts of analyses, we may actually recover some interesting and unexpected sources of diversity in lineages that are otherwise kind of unexciting for how much phenotypic diversity they have. But I would fall short of saying necessarily that it's going to be the case that all non-adaptive radiations are actually adaptive in hidden channels. May or may not be.
All right. Well, thank you for a great talk and a great discussion. Now until 2 o'clock, there's a lunch with the graduate students. If you can remain here.